Good morning and good afternoon. Thank you for joining day 3 of our 2020 virtual user meeting, ligand-gated ion channels in drug discovery. My name is Jason Villagomez, Marketing Manager at Nanion Technologies, and will be your moderator for today's presentations. By default, all participants are muted, and upon completion of the presentation, I will open up the floor to some questions. We encourage you to submit your questions throughout the course of each presentation. It is now my pleasure to introduce Dr. Marc Rogers, Chief Scientific Officer, Metrion Biosciences from the UK, presenting development and validation of ASIC-1A ligand-gated ion channel drug discovery assays on automated patch clamp platforms. I'll now hand over the conversation to Marc.
Right, thanks, Jason. Hello, everybody. Good morning, good afternoon, good evening. So it's always a pleasure to talk at these events. Thanks to Nanion for inviting me. So we've been using the Nanion platforms for well, a number of years, actually decades. So I'll give you an overview of what we've been doing, mostly on the patch liner, but also some early work on the Synchro 384, with a focus on ligand-gated experiments, and in particular, the ASIC-1A ion channel that we've used as an exemplar for our experiments. So just a quick overview. So I'll introduce Metion very briefly for those of you who are not aware of us. I'll give it a quick check on what we've been doing with the patch liner early days, which is mostly voltage-gated work. But now we're moving into the space of ligand-gated receptors. And I'll to take you through 3 different ways that we've used the ASIC-1A cell line on the various Nanion platforms to develop drug screening assays for ligand-gated receptors. So traditional sort of pipette-based systems, the stack tips that are available on the patch liner and the Synchro, and also the last couple of slides will be an old school example of some classical biophysical techniques that were used in the past to study ligand-gated receptors and native cells, but that we were able to transfer onto the automated patch clamp platform. So Metrion Biosciences is based in Cambridge, UK. We're an ion channel specialist CRO. We've set up a core expertise in cardiac safety and neurotoxicology screening, also ion channel drug discovery screening. So we use a combination of heterologous cell lines, neuroblastomas, and also native neurons and stem cell derived materials for our drug screening services. So our early work with automated patch clamp, and in particular the Nanion patch liner, dates all the way back to perhaps 2008, I believe. So Michael and Cecilia actually came over and installed our patch liner. And we focused really for almost a decade just on voltage-gated channels. Obviously, the first one we set up on the patch liner was the HERG, go figure. So our safety pharmacology assay, so you can see that on the left-hand panel there, just an example from our 4-channel patch liner, 3 cells showing HERG tau currents. But the main use of the patch liner over the last decade or so has been for 2 voltage-gated ion channels that are involved in pain. So for analgesia drug discovery targets. We had a project with a Japanese pharmaceutical company looking at NaV1.7 and NaV1.8, as you can see here in the middle panel. So we're looking at state-dependent blockers. So we're looking at resting state and then inactivated state channels and screening test compounds. So that was a 3 or 4 year project. And when that finished, we pivoted to a CAV 2.2 N-type calcium channel pain program with Grunenthal. So this was an 8-year collaboration. And in that time, we were doing the primary electrophysiology screening to support the MedChem SAR, and also the gene selectivity profiling for the other neuronal calcium channels, so the PQ type and the T type. So there was a lot of work that we did on the patch liners, but all of it was focused around the voltage-gated, and we're very happy with the patch liner for that, and in particular, the use of the metal tips for the compound applications. So you see in the bottom right-hand corner here, an example of a 4-point cumulative IC50 testing of a lead compound, so showing the inhibition of the CAV 2.2 channels. Our use of ligand-gated receptors on the patch liner was actually much more limited, and our expertise and our experience over the last decade or so has really just been focused on trip channels. So those of you who've been looking at the other talks today will already have seen quite a lot on various trip channels on the Nanion devices. Our initial focus was using TRPA1, and then we also had a small project on a TRPM channel. And the way we approach these, because they're slowly activating ligand-gated receptors, so we would use voltage ramps. So apply the agonist for a slowly acting, non-desensitizing concentration of agonist. For example, on the left-hand side for TRPA1, we use cinnamaldehyde. And then we would run a voltage ramp before and after the presence of agonist. And this is both so we can actually look at changes in the seal and do QC, but we're also potentially interested in antagonists or agonists that have a voltage dependence. So trying to extract additional information from the automated patch clamp assay during the ligand-gated experiment. So we would apply a voltage ramp from negative to positive potentials every minute or so in the presence of agonist. And you can see here that you get an increase in the inward and outward currents in the presence of agonist. You can then create an EC50 curve for activation of the receptor. And on the right-hand side, we've got a little bit more complicated assay for the TRPM. Same thing, voltage ramp with and without agonist. And you can see on the time course on the right here that you can get a fairly well-sustained current during the continuous presence of agonist. You can wash the agonist off and then you can apply it again. So you want to make sure that you have a stable response with these TRP channels. But these are fairly slow activating. This is kind of an intermediate type of assay between a voltage-gated assay and a ligand-gated assay. And what we've been seeing in recent years as a CRO is an increased number of requests from clients to look at what you call classical neurotransmitter-gated ion channels. So GABA-A, glutamate, P2X, and nicotinics. So this is a bit of a challenge for us. So we have to really move into full ligand-gated assay mode. And our previous experience, and those what many other people started with these automated patch clamp platforms, would be to use your traditional metal pipette. I'm calling it the pipette tip-based system. So the left-hand graphic here, you would have pick up the agonist with or without antagonist in your pipette, and then move the pipette over the cell, squirt on the agonist. Then you'd have to remove the pipette, go and get some wash solution, bring the pipette back to the cell, and then wash off. So you see during this 10-20 second experiment that you've got activation, but you've also got desensitization, and you have to wait quite some time until you can get the wash solution and then have a return to baseline. And this is fine for some receptors, but many of these classical ligand-gated receptors are subject to some quite severe desensitization. So the question is, how stable would an assay be, and would it be suitable for drug discovery screening using this type of technique? So what intrigued us was the ability to do stack tips on the Nanion platform. So starting off with the patch liner. And the idea here on the right is you can actually compile your ligand-gated experiment within each tip. So you will first of all take up your wash solution, and then you would take up agonist, and then you would take up some more baseline saline, and then you'd move the tip over to the cells, and then slowly or rapidly apply this whole ligand-gated experiment through the tip onto the cell. So you see you have a nice baseline, then you've got rapid activation, you have some sustained current during the peak, and then you have a nice wash off back to baseline. And the time course of this experiment is significantly faster than that you see with a traditional pipette-based system. So we were obviously very keen to set up these assays using patch liners. And we decided to use ASIC-1A as our exemplar ligand-gated receptor. Now, this is a validated drug discovery target. It's implicated in ischemia damage. Obviously, it's activated by pH. So this happens during tissue damage, both in the heart and in the brain. And also, it's one of the suite of what we might call metabolic receptors and sensory nociceptors, such as the TRPs and the ASICs that respond to tissue damage and inflammation mediators by exciting the neurons and producing pain. So there's a number of different receptors and subunits for the ASIC family. They're an interesting sort of subtype because they're a trimeric protein. So you can either have homomeric or heteromeric combinations, and there's also some splice variants, hence the ASIC-1A is a splice variant. There's also an ASIC-1B receptor. And a number of these are actually implicated in pain because they're expressed in the sensory nociceptors, and they respond to both to increased pH. But it's quite interesting that there's also pretty typical for ion channel drug discovery that there are some animal toxins that have evolved to target these receptors. So there's a snake toxin from the Texas coral snake that actually activates ASIC1A, and that's part of why it's so painful for that snake bite. And there's also a number of toxins from different species that are antagonists of the ASIC1A. So there's the Sama toxin and also the mambalgins. And both of those have been subject to various drug discovery programs. Sama toxin has been developed by University of Queensland group for drug discovery, and there's also a French group that were trying to develop mambalgins and analogs as analgesics. And you see on the right-hand side here that there's a classic kind of neuropathic pain readout here, von Frey, mechanical allodynia, and you get a reversal of this pain with the Sama toxin, about 50% of the value of morphine. This is very significant because this is a novel mechanism of action. This is non-opioid. This should be non-addictive. There's a big push on to have non-opioid analgesics come to the market. So having novel mechanisms of action, such as targeting ASIC1A or CAV2.2, they're of high value and high interest in the drug discovery field, especially for pain. And this is some data from the Australian group. So they're putting the Sama toxin into animals that have got a cerebral infarct. And you see that the Sama toxin reduces the size of the infarct in the brain. So this is a potential treatment for ischemia, and they're taking this through all the way through to the clinic. So ASIC1A is a good test bed, a good exemplar for ligand-gated assays, but it also has direct relevance for drug discovery screening. So there's been a number of studies by other groups. I was making it really clear this is not our data. I've just found this on the Nanion website. So people who've looked at ASIC1A responses both on the patch liner on the left and also on the syncropatch on the right, using the traditional type of pipette-based system where you've just got agonist in the pipette and then you apply that onto the cells. And what you see in both cases is you've got this transient response, but there's also a monophasic decay. And what's happening is the size of this current is decreasing or decaying during the application period. So this is not wash off, this is desensitization. And this happens both on the patch liner on the left and also on the recordings from the syncropatch on the right. So this is, I'll just give a big shout out to Nina Braun. So I think this is from her PhD study. So in both cases, the prolonged application of agonists produces this desensitization. This is not really a problem for the ASIC channel itself because it has rapid recovery from desensitization, but this would be a problem for getting a stable assay suitable for drug screening for many other types of classical neurotransmitter receptors such as nicotinics and NMDA and even for GABA. So this standard kind of pipette-based, tip-based application in our mind is not suitable for drug screening and it may not be suitable for receptors other than ASIC1A. So we were interested in looking at other types of ligand-gated approaches. And this is where we started working on the stack tip with the patch liner. So there's a schematic on the left-hand side, which illustrates how it's all set up on the patch liner chip. So you've got the pipette tip on the left-hand side. So you've taken up wash-based or saline solution, and then in blue it says drug, but really we should look at this and say we should call this agonist. So again, you've taken up a whole ligand-gated experiment into the pipette. You then place the pipette in the recording system, and then you flow the contents of the tip across the cell, and then you get a ligand-gated response. So you get a rapid activation, you get some peak current, and then you get the wash-off as the saline comes across the cell, and you get a nice kinetics for a ligand-gated experiment. And what's particularly interesting to us, and I think particularly powerful for the stack tip technique, is you can change the way your experiment is designed within the tip. So you can have different volumes of agonist and wash. You can have different timings that they are applied to the cell, and you can change the speed at which the experiment is flowed over the cell. So on the right-hand side, you can see an example of what we might call a slow stack tip. So you've got rapid activation, but then you have a profound period of desensitization. That is potentially interesting from a drug discovery perspective. But then when the wash solution hits the cell, you then clearly get a rapid return to baseline. And if you speed up the flow of solutions over the cell, then you can get a faster ligand-gated response, where your responses begin to look more like a synaptic type of response, which is appropriate for a neurotransmitter assay. And so you get a lot of flexibility with a stack tip. And on the bottom right-hand corner here, this table, just an example of the settings that we got out of the box from Nanion for the stack tip on the patch liner. And then after some optimization, these are the parameters, the speeds and the volumes that we used for what we thought would be an optimized ASIC 1A response on our patch liner. And the other thing about this system that I should mention, of course, is you're putting the whole experiment within the tip into the recording site, but then you can pause. So what's also important here is as you're moving tips in and out of electrophysiology recording wells is you potentially get electrical noise and you get artifacts. So you get these liquid addition and pipette entry artifacts. You can separate that artifact from the actual ligand-gated experiment with the stack tip. And you can see here just an example. So the tip goes in here, there's a small artifact. We can wait for that to dissipate and then we can start the ligand-gated experiment. So you get a nice clean recording and you can get accurate recordings of the rapidly activated peak current. So there's some real flexibility and some power in the stack tip technique, which is very useful for ligand-gated experiments. So this is an example of our optimized ASIC 1A response on the patch liner. So we've got some nice, we've got a good period of baseline. We want to make sure that we don't have any leaky cells. We then apply the agonist, we get rapid activation and a small amount of desensitization. And this is not a problem. We actually want to see some desensitization. This is the normal functioning of a ligand-gated receptor. This also tells us that we know that we've got the maximum peak activation under those particular conditions. With the previous traces you saw, you had this onward and off, on and off rate, and it was kind of hard to be absolutely sure that you've got maximum activation and we're measuring the true peak of the response to the particular concentration of agonist you were applying. So we like to see a response like this, which is actually quite physiological. But then when you go to the wash solution, you see a more rapid, so there's a biphasic decay here. The first phase is desensitization, the second phase is wash off, much more rapid, and you get a complete return to baseline within the whole experiment lasts about a second. And that's exactly what we want. And with the complete wash off here, you can then repeat the experiment every minute or so, so you get a nice stable baseline under control conditions, because you will eventually be testing compounds against that response. So you want to have at least a good 20-minute of stable responses to the ligand-gated application. And where you start off with all ligand-gated experiments, of course, is estimating the EC50 or EC80 of your agonist. So here's the patch line at EC50 for pH activation of ASIC 1A. So we get a EC50 of about pH 6.6, which is exactly spot on in terms of literature values. They usually go from between 6.4 to 6.6, 6.7. So this was exactly what we wanted to see. That's setting up the assay. We then wanted to create an assay that would be suitable for drug screening. So basically to put on either positive or negative modulators or antagonists. In the case of ASIC 1A, we're looking for either NAMs or straight out competitive antagonists to reduce the inflammatory response and to be potential analgesics. So it gets a little bit more complicated for antagonist screening and ligand-gated, but the stack tip is really quite perfect for this. So the schematic on the left-hand side, each of the purple bars is where we're applying a stimulatory concentration of pH. In this example, it's pH 6.5, which is about the EC50. We get 3 applications here, along with the gray, which is in baseline, so make sure we've got a stable response. But what you can do with the subsequent applications here is, instead of having saline as the wash-off, you have the next antagonist concentration as your wash-off. So essentially, you're pre-incubating the cell in antagonist, and then you're flowing on antagonist plus agonist. And then the final application within the pipette, the stack tip, is the next antagonist concentration. So in each of these experiments, we're pre-incubating with antagonist. And it's really easy to do that with the stack tip. And in this case, we're showing you the IC50 for one of the non-selective ASIC antagonists, benzamyl. So we get an IC50 here of 3.5, 3.6 micromolar, which is again completely in line with literature values. And I'll refer back to this because we've got some other benzamyl data from the other platforms and applications later on in the talk. So this is again the additional advantage of the stack tip, is you can design a drug screening antagonist profiling experiment within each tip. And you can apply exactly the same experiment to every cell because it's predetermined by the liquid you put into the stack tip. So you have very similar timings and similar results when you use the same tip against different cells. The issue, though, is you are limited by how much work each tip can do on your automated patch clamp platform. So with our 4-channel patch liner, we've only got one pipette tip. So we can only do one cell at a time, and then the other 3 cells are waiting around. They will get the same experiment, but they're going to get it not in parallel, it'll be in series. So you're really not utilizing the full throughput of your automated patch clamp system if you are limited to doing ligand-gated, even stack tip experiments in a pipette-based system. So really, it's obvious that if you want to increase your throughput, is you need to look at the 384 well systems. And the real beauty here, the real power here is, of course, it's 384 tips onto 384 cells. So every single cell receives the same experiment at the same time. None of the cells on the right-hand side of the chip are waiting around for 20 or 30 or 40 minutes to contribute their data while they wait for the ones at the front of the chip to actually do the experiment. Everything is done at the same time. You get this massive increase in throughput, but you also get a major increase in the consistency of the assay across the plate. And this is really important when you're testing compounds. You want to make sure that response you get from compounds that are applied to this part of the chip have a similar chance of operating against the agonist responses as compounds that are added right at the end of the chip. So we looked initially at the SynchroPatch 384, and I should go back to what I said before. We come from a voltage-gated background and compound screening, and there's way too much literature and discussions and posters and discussions about the issues with applying compounds with plastic tip-based systems. You get compound absorption, you get shifts in potency. From a compound screening point of view with voltage-gated systems, there is a certain resistance, and I would certainly put my hand up and say I'm very much a doubter about the efficacy of these systems with all of the plastic in the system. We've gone to a lot of trouble at Metrion to get rid of the plastic in our automated patch clamp screening. But I think there are certain advantages, and perhaps the advantages definitely outweigh any potential disadvantages with a plastic tip system when you've got this full throughput capability for ligand-gated. And add on top of that, you've got the ability to do stack tip experiments on the 384 systems. So we finally decided that we should have a look at these systems. So Nanion staff were kind enough to invite me over for a demo late last year. And so most of the data and the rest of the slides come from the 384 demo that we did with the ASIC 1A cell line. So this is a schematic showing the various options that you have with the 384 system. So we've got something similar to the pipette-based system where you just have agonist in the bottom of the pipette. Nanion folk call this a puff, and they quite like this technique. What we wanted to try with the stack tips, and a standard kind of stack tip here is where you again have a small volume of agonist and you have a large volume of wash, and this is your wash off. So you're trying to get rid of the agonist off of the cells. And what's turned out quite interesting and counterintuitive is actually this system, this sort of layout doesn't actually work as well as you would think. And it's not just in our hands, but also I saw some nice data from David Dalrymple and the GABA-A assays they set up on the SynchroPatch. They started with these standard stacks and they didn't actually get good wash off, and they did see desensitization. I think our understanding here is that rather than completely washing off the ligand, you're really just diluting it in the well system, the small well system. And so there's a mixture of wash off and desensitization. So with a standard stack, you don't get a complete and rapid return to baseline. And rather counterintuitively, if you reduce the volume of the wash, you actually get a better response. So we're calling this a short stack. And I think the idea here is because the tip stays over the cell in each recording chamber, you get a localized wash off of the agonist, but it keeps the bulk solution away from the cell. So you do get it on a micro scale, on a cellular scale, you get a more effective wash off. So on the right-hand side here is a SynchroPatch recording from our ASIC demo. So this is the puff technique. And you see during the application of agonist, you do get a peak, but you do get wash off / desensitization. And then it takes a long time to get back towards baseline. So that's not what we were looking for. What we're looking for is on the right-hand side with the short stack. So we've got a more rapidly activating and profoundly desensitizing response because we've got a more like the EC80 rather than the EC50, pH 5.5. But crucially, when the agonist comes off and then that small wash volume here is applied, you get a complete and rapid return to baseline. And that allows us to have repeated responses. And this is exactly the kind of profile that we're looking for in a ligand-gated assay on an automated patch platform. So this is just a comparison again of the single puff versus the short stack. Most importantly here, because we're going to be doing drug screening, we want to make sure we've got a stable response over time. So on the left-hand side here, you've got repeated applications of pH 6.5 using the puff. And actually, even though you have these slow kinetics, they're very reproducible. And if you look at the current amplitudes here, you've got our 3 baseline recordings, and then you've got 4 other recordings that are done in DMSO. And on the right-hand side, we would repeat this experiment, but with increasing concentrations of test compound or antagonist. So this is we get good stability and it allows us to do screening. I think why this looks so good and why it works so well with the ASIC is that it's particularly good at recovering from desensitization. So it has a relatively fast off rate for the hydrogen ions, the pH. I think you might have more problem getting a stable recording with a desensitizing current such as alpha-7 nicotinics. And I know there's a talk later on today where Omar will be talking about some nicotinic assays he's running on the patch liner. On the right-hand side is our preferred method. So this is the short stack. And again, we've got rapidly activating. We know where the peak is. We can see the desensitization. This is what we want to see and expect from an ASIC response. But we get repeated and rapid recovery to baseline so that we then get a nice stable recording, which would allow us to do drug discovery, compound screening. So in the middle here, we've got a short stack EC50 curve. So again, here we've got peak, then we've got desensitization, then we've got rapid wash off. So for this experiment on the synchro, we've got an EC50 of 6.44. And if you remember, EC50 on the patch liner with stack tips was 6.6. So very similar results, but an incredible increase in throughput and consistency. So we were very happy with that. So we then went on to testing our reference compounds. So again, I might have failed to mention that ASIC is a member of the ENaC and degenerin family. So they are sensitive to these non-selective blockers, amiloride and benzamyl. So these are small molecules. This is similar to the type of small molecules that many pharma companies and biotechs are developing as antagonists and modulators of ASIC1A. So using the puff here, we get an amiloride IC50 of 6 micromolar, and a benzamyl we get 1.36. So again, if you recall, on the patch liner, we got an IC50 of 3 micromolar for benzamyl. So same, we get really good responses. And in on the table on the right-hand side, we've repeated these experiments with the short stack. And again, you can see really there's no significant difference at all between the IC50s for these reference compounds using the 2 different ligand application techniques, although we would still prefer to use short stack, possibly or probably for other ligand-gated receptors that are not so capable of recovering from desensitization. But in terms of establishing pharmacology, we were really happy with this result. And of course, you get massive throughput. And this is just an example of the synchro patch throughput that we can achieve and the success rates. The point here as well is that there are additional flexibilities and options available on automated patch platforms to optimize your assay. So we prefer usually to start out with single hole assays because we like to be able to QC each and every cell. And we're looking at the QC rate here in the table. This is not just instantaneous success rate. This is not just seals greater than 200 megohm or whatever. This is seal quality, seal stability over the course of the experiment, series resistance, and also current amplitude. So this is a full fat success rate, QC success rate. So in a single hole, conventional whole cell recording, we get a success rate of 81%, which is tremendous. There's most of the failures are in gray or through low expression, and there's a couple of poor seals in blue. It's really important to mention that we're getting genuine gigaseal recordings here from 384. This was not always the case with some of the earlier 384 platforms. So this is a really powerful technique here. But it wasn't enough for the Nanion folk. I mean, 80% success rate for us, that's pretty good. But they wanted more. So Marcus and Tom and others pushed us a bit harder. So a lot of people use multi-hole chips, and certainly on the right-hand side, you can see that if you use a 4-hole chip, you can get rid of the failures from low expression. So we're now up to 94% success rate. There's just a few cells still that don't have sufficient current and a couple of poor seals. But the seal quality with the 4-hole is pretty much the same as the single hole. So if you multiply this by 4, you've got a 1.2 gigohm average for the single recording site. And if you look across the whole plate here, so we're doing a screening mode. We've got our positive controls and negative controls, and then we've also got our reference compounds. So we're doing a whole column of 4 columns per compound. Obviously, if we was in drug screening mode, we would block this out and be able to test a lot more antagonists in each run. And we'd be able to do that all at once, and every single cell would receive the same experiment using the stack tip. So our plate-based metric here, assay metric, Z prime is around 0.75. So we've gone from a single cell at a time, work your way across the patch liner chip, to all-in-one big bash, 384 cells all at once. So we're getting this data, a lot more data, obviously a multiplier of what we can achieve on other platforms in the same amount of time or a shorter amount of time, with great success rates and really good assay metrics and QC parameters. So we were pretty blown away by this. Clearly, there is a tremendous capacity on the Synchro 384 for doing studying ligand-gated receptors in a drug discovery screening mode. So that's the main focus of the talk. I've got a couple of cheeky slides at the end here for some old school biophysics. So I'm older than I look, I should mention. So I did my PhD in the 80s, and I'm a neuroscientist by training. And a lot of the early work on ligand-gated receptors were looking at neurotransmitter responses in native neurons, in slices or even in vivo. And for those of you who are far younger than I am, you might not remember this or be aware of this technique. So if you've got a neuron and it's sitting in a bit of the brain, you really don't want to be doing voltage clamp, because you don't have good space clamp. You've got all these dendrites and axons. And most importantly, actually, you've got quite a lot of your neurotransmitter receptors are not on the soma. They're out here on the processes where the synapses are. So a more reasonable approach to study these receptors early on, pre-wholesale patch clamp, pre-1980s, was to do current clamp recordings with sharp microelectrodes and then have an interferesis, so have a puffer pipette that you would move over the brain slice, and then you'd puff on your agonist, your neurotransmitter. So this is an example trace here. So if you've got a receptor that is rectifying, you're holding it, the cell is sitting at negative membrane potentials, and you're current clamping the cell, and then you apply on an excitatory neurotransmitter, then you're going to get a depolarization here. And this mimics the EPSP, this mimics the synaptic potential. And current clamp, that's how the brain computes, that's how neurons work. They don't work under voltage clamp, they're under current clamp. So this is a classic kind of interferesis ligand-gated experiment. The additional detail here, you see these jumps, these steps. So under current clamp here, you're also injecting current steps, and these are measuring the input resistance of the cell. And during the application of agonist, you are decreasing the resistance because you're increasing the conductance of the cell when the ligand-gated receptors are open. So the size of these current steps or voltage excursions is reduced during the agonist application. And the same thing would happen if you were positive to the reversal potential of the channel of interest. You would, instead of a depolarization, you get a hyperpolarization, but again, you get a decrease in the size of these steps. So I thought, well, you've got current clamp facilities on quite a number of the automated patch clamp platforms. Would it be possible to carry out this technique, to adapt it to automated patch clamp using a heterologous cell that's expressing a ligand-gated receptor? So this was a Friday afternoon project, as we call them at Metrion. I gave this to Sam Manyweathers. He presented some of this data at the EU user meeting, I think 2 years ago. And he was, after some considerable effort, able to make this technique work on the patch liner using the current clamp facility. So we've gone from a technique that was used in the 60s and 70s and the 80s with microelectrodes in the brain to actually being able to show that we can do it on automated patch clamp using stable cell lines with high throughput. So the same thing here. This is just the example of the method we use, the biophysical method. So we're looking at here at current clamp, we're looking at voltage. We're putting in these current steps, current injections that give you a voltage deflection. Sam sets the assay up a little bit by using a ramp. So increasing current injections give you increasing size of voltage deflections. So this is a measure of the input resistance or the conductance of the cell at rest. And then he repeats that in the presence of pH activation of the ASIC-1A channel. And as we saw on the previous slide, these voltage excursions are smaller in the presence of agonists because the input resistance is decreased and the conductance is increased. So if you plot this change against pH, you can get an EC50 curve that looks pretty much the same as we get from voltage clamp and fast ligand application with pipettes or stack tips. So we've got an EC50 here of 6.68. So very similar to what we got with this traditional technique. So this is really just a bit of a play, but it does tick the box in terms of showing that you can develop and validate several different ways of looking at ligand-gated receptors on automated patch clamp machines. This probably wouldn't be your first choice of technique for drug screening, but I thought it was a very powerful demonstration of the flexibility and the capability of automated patch clamp machines. And people need to, I think, you can get some real advantages by looking at all the capabilities on the machine and trying to push the envelope out a little bit, and also trying to take historical information and apply that to 21st century screening platforms. So I was really pleased. I do thank Sam for his effort on this project. So just to wrap up, so we've successfully used ASIC-1A as an exemplar ligand-gated target to develop assays on a number of automated patch clamp platforms from Nanion. We're a particular favorite of using the stack tip techniques, which we find to be very powerful and very flexible. And it is a way for you to get optimized ligand-gated assays that are suitable for drug discovery screening. The patch liner, being a medium throughput device, is very good for assay development, and we can get very good translation of assay conditions set up on the patch liner onto the high throughput 384 synchro patch. And we have the same capabilities on the synchro as you do on the patch liner. We've got the stack tips, and we've got the current clamp as well. But what we really gain there is the fact that you've got one-to-one correlation between the tips and the cells. So you can get some a huge increase in the consistency and the throughput of your ligand-gated experiments using the synchro. And there are additional flexible options, of course. You can also use partial plates, partial chips for assay development. And then you can scale up to the full chip. You can use either single hole or multi-hole. You've also got the options of different resistances for each of those hole options on the Nanion chips. And you can also use either conventional or perforated patch recordings to further optimize your experiments. And finally, you can use the current clamp facility to devise alternative biophysical screening assays. So that's me done. The most important slide, of course, is the acknowledgements. So there's quite a number of people who worked on the patch liner over the last 10 years or so, 15 years. So Rich and Neil set up and ran the sodium channel assays. Rob and Kathy ran the CAV 2.2, and then quite a number of other Zentian and Metrion employees are involved in that project. Tony and Ian and Ray set up and ran the TRIP experiments, and Sam did the ASIC-1A. And on the right-hand side, it's not everybody at Nanion. Of course, there's a lot more people at Nanion now than when I started working with Nanion 12 years ago or so, but these are the important people who helped us with those various assays on the left-hand side. So I thank Nanion staff for their support, and I thank everybody for their attention. And hopefully there's a few questions, Jason.
Hey, Mark, thanks for going over that presentation. It's really great. So yeah, let's move on into the Q&A. We've received some questions already. If you have any additional questions, just feel free to type into the chat and we'll try to get through them. If not, then we will make sure to have a transcribed copy. Going back to the beginning of the talk, Mark, you were talking about the tips, and then we had a question come in regarding any alternative to plastic tip systems, metal tips?
Yeah, so we used a couple of different types of metal tips on the patch liner. So there was early days, there was some we had glass coated, we've got Teflon coated. There are various options, and certainly it's good to ask the question of Nanion staff and also some of the other users, what experience they've had. A related question is, at the moment, I think most people are using the standard, on the Synchro, are using the standard tips that come with the liquid handling device that the Synchro is built into. And our demo with the 384i was using the new Biomech, but just the standard tips. So I think there actually is potentially some gains to be made if you're going to be looking at low compound absorption plastic tips for if you're going to be using these platforms for drug screening. So I'm not aware, I haven't done the research. I don't know what the options are. I'm sure you could get other 384 tips, and you could probably do those experiments yourself to try and see whether there is compound absorption, how bad it is, and whether you can reduce it by using different types of plastic tips. But again, both systems offer flexibility in the composition and the type of tips that you can use.
And follow up to that, do you think that there's a lot of absorption into the plastic tips, given that the compound is only in the pipette tips for a few seconds or perhaps up to a minute or so?
I can't answer that because we didn't go to that level of experiment with the Synchro. Obviously, that would be one of the set of experiments that we would like to do. So we have a great deal of reference pharmacology already from metal pipette tip-based systems for voltage and ligand-gated channels. So we would compare that reference pharmacology with what we saw in the Synchro. Certainly looking at all the Synchro data, and there is a lot out there, I've not seen or noticed any real significant potency shifts for a wide range of ion channel targets that have been tested by Nanion and Synchro users. So it doesn't seem to be nearly as significant as it was in the early days. A lot of this came out of the ironworks, and there was just plastic everywhere in the ironworks, and there's a lot less of it now in the modern systems. But it's a learning that doesn't go away. There's some old farts like me that it's still very much in our mind. Maybe we bang on about it a bit too much, but it is an issue, and I think it's good for people to test whether they are having that issue and to try and address it rather than just ignore it.
And then regarding the pre-incubation, how long was the pre-incubation and the length of, how long the pre-incubation was, and if the length of pre-incubation makes a difference in the IC50?
We had a fairly standard, so the experiment was mostly designed around getting a stable response. So we would have between 1 and 2 minutes between each application of pH. So by default, that was the pre-incubation period. I should say on the voltage-gated work that we've done, we typically would do a 4-minute incubation with compounds. So we would have two 2-minute applications of compounds while we were doing the voltage protocol. So with the ligand-gated, we're probably cutting that down to about 2 minutes. But again, you have the flexibility. You can leave the compounds pre-incubating for as long as you want, and then reapply your pH. So again, you have the flexibility, not within the tip, but within the experimental scheduling for that. So in those examples, it was a 2-minute pre-incubation.
And then is it vital to do assay development on the patch liner before transitioning to the work that you did on the Synchro patch, or can you reliably do assay development directly on the Synchro patch?
I think you can definitely do assay development on the Synchro patch. It's just the way that we work and the fact that we have a number of patch liners at Metrion, so that's where we start. And we don't yet have a 384 device. So we do the development in-house, and then we go and test out the assays at other sites and with other partners and demos, for example. Certainly, I think that what's really powerful for the Synchro is the partial chip. If you can use, obviously you don't want to use a partial chip and then not use the rest of it until a week later or whatever, but you can spread it across a day, you can spread it across a couple of days. We did this on the demo. I didn't see any decrement in performance using a used chip. And I think that's a really important aspect in terms of assay development. Some people just jump in and they get 50% success rate and they're really happy with that. But I think you really should aim a little bit higher for that. And certainly for us in the CRO setting, we want to have the most efficient assays as possible.
Yeah. And then another question, if you've tried stem cell derived neurons on the patch liner or if you plan to do so.
No, we did stem cell cardiomyocytes back in the day. We've not done neurons. We've had a few clients who've talked to us about that. I'm not sure what you would gain on a patch line, but certainly if you were to put stem cell neurons onto 384, and we've seen some people, we saw some talks like a BPS and a user meeting. So our Kermes have done that for Toe. She's not there anymore, but they did it, and a couple of other groups have done it. I think the advantage, there's a disadvantage as you lose some of the physiological composition of the cells and the morphology, especially with neurons, if you put them on automated patch, because you have to dissociate them. But you gain a massive amount in terms of data sets. So if you were interested in the expression of a particular ion channel in your stem cell neurons, then trying to do it one by one on manual patch or patch line is going to take a long time. You still only have a limited data set. Being able to do it on automated gives you that chance to get 100 cells and have a really powerful statistical analysis of current expression. And again, people are using the synchro patch to look at mutations, for example, patient-derived mutations. They're expressing the ion channels in heterologous cells and looking at biophysics. But I think there's a real application for, again, comparing the current expression, current density, biophysics of control versus patient-derived mutations that are expressed in stem cell neurons, for example. And for that kind of statistics, you need to have, 60, 80, 100 cells. So what you might lose in terms of physiology, you gain in statistics.
And then lastly, just in closing, what are your 4-point QC criteria for the SynchroPatch 34i experiments? How long did the cells last? Yeah, if you don't mind answering.
Oh, it was quite an intense demo. I was really impressed. I mean, I'm quite hard to please. Those of you who know me, I'm quite hard to please, and certainly in terms of ion channels and electrophysiology. And I grew up with the Barracuda and the Ironworks, where they were not great recordings. I was really blown away by the quality that we got, and it really is like for like compared to other automated patch count platforms that we've used. So I think it's fair to say we were asking for more rigid and more stringent QC parameters than are normally used at Nanion. So we were asking for lower series resistance, higher seals, and we weren't just taking those stats at the beginning of the experiment, we were also looking at them in the middle and the end of the experiment. And we had all of those. Again, you've got the power of the data control and the software that you can set those parameters, quite a number of parameters, either in isolation or combination. So we were looking for at least 200, 300 megohm as the minimum seal, and then series resistance less than 12, say 12 megohms. And we wanted to see those throughout the course of the experiment. So these are between 12 and 20-minute experiments. So I think that's a fairly reasonable real-world expectation.
Perfect, thank you. Well, once again, Marc, thanks for going over the presentation. We'll make sure to provide a transcription of everything. Also, the on-demand copy will be available by tomorrow morning at the latest for those on a different time zone that maybe were unable to join live. And if there's any additional questions that you'd like to ask to Marc, I'll make sure that his contact information is also in the description, as well as all the folks from Nanion who are more than happy to answer any outstanding questions you might have. Thank you for joining our talk. The next talk will begin at 4 p.m. Central European time, 10 a.m. on the East Coast, and we hope that you can join.
All right. Thanks, Jason. Thanks, everybody, for